An alcohol can react with phosphoric acid to produce a phosphate ester or phosphoester, shown down here. The phosphate ester can then react with a second or third acid to give phosphoric acid anhydrides, which are seen in molecules like ADP and ATP. ADP and ATP of biochemistry fame are important examples of phosphate esters. When two phosphate groups react with one another, a water molecule is lost. Because water is lost, the resulting bond is called a phosphoanhydride bond. A phosphoanhydride bond is the same thing as a phosphoric acid anhydride. Adenosine triphosphate, or ATP, is the universal energy currency for all living organisms. Much of the energy harvested in cellular metabolic reactions is stored in ATP molecules. The ATP molecule consists of a nitrogenous base, adenine, and a phosphate ester of the 5-carbon sugar ribose. Here's ribose, and here's my phosphate ester. The triphosphate group attached to ribose is made up of 3 phosphate groups bonded to one another by phosphoanhydride bonds. These phosphoanhydride bonds are termed high-energy bonds because their energy is released when they are broken. Thus, the metabolic energy stored in these high-energy bonds can be released for use in energy-requiring reactions. Cellular enzymes can carry out a reaction between a thiol and a carboxylic acid to produce a thioester, shown in the box on the top right. The reactions that produce thioesters are essential in energy harvesting pathways as a means of activating acyl groups for subsequent breakdown reactions. The complex thiol coenzyme A is the most important acyl group activator in the cell. The detailed structure of coenzyme A appears in Chapter 12, but it is generally abbreviated CoA-SH, like this, to emphasize the importance of the sulfhydryl group. The most common thioester is the acetyl ester called acetyl coenzyme A. Acetyl coenzyme A carries the acetyl group from glycolysis or beta oxidation of a fatty acid to an intermediate of the citric acid cycle. This reaction is an example of an acyl group transfer reaction. In this case, the acyl group donor is the thioester acetyl coenzyme A. The acyl group being transferred is the acetyl group, which is transferred to the carbonyl carbon of oxaloacetate. This reaction, which is catalyzed by the enzyme citrate synthase, is shown here. Later on in this course, we will see that the citric acid cycle is an energy harvesting pathway that completely oxidizes the acetyl group to 2 carbon dioxide molecules. The electrons that are harvested in the process are used to produce large amounts of ATP. Coenzyme A also serves to activate the acyl group of fatty acids during beta oxidation, the pathway by which fatty acids are oxidized to produce ATP.